So welcome back, everyone. We have 5 talks over the next 45 minutes. There will be questions at the end of talk number 1, as it has a 15-minute time slot. And then the following 5 talks are 5-minute lightning talks. So we'll hold the questions for those speakers until the end, and you're welcome to put any questions you have for those speakers in particular in the chat as well. So we're going to start off first off with Carter Feltham, nesting dynamics in space and time in a population complex of blandings turtles in Nova Scotia. So Carter, if you want to share your screen.
Thank you so much for inviting me today. So as Carla said, the nesting dynamics in space and time in a population complex of blandings turtle in Nova Scotia. For a quick introduction, I represent the Mersey Tobeatic Research Institute, or MTRI for short. We're located in the South Shore of Nova Scotia, and we're plunked pretty central in the middle of these blandings turtles, which occur in a disjunct population, and there are 4 known subpopulations within this population. And we've had a volunteer nesting program for about over 20 years now, and this paper was based on that data. The methods used here, we looked at the nest protection data from 2003 to 2022 in 3 subpopulations, so SP1, SP2, and SP3. As there's no active nest protection in SP4, it was only discovered in 2016. We examined trends in clutch size, nesting frequency, seasonal timings, hatching success, recruitment, and age of maturation. Most of the data was analyzed in R, primarily using linear mixed models, general linear mixed models, and general additive mixed models. So from this, we looked at 870 protected nests. There were some other nests here that were not included. Some of them were collected for incubation or head starting programs. Some were predated. There were a few that have no eggs. We actually have a ghost nest turtle who goes through the entire process but doesn't actually lay any eggs. And of these, 654 produced hatchlings, 198 failed, and 18 were of unknown fate. So how many eggs do blandings turtles lay? Clutch size was significantly influenced by carapace length. So the larger the turtle, the bigger the clutch size. And it was also influenced by subpopulation. So they actually significantly were different between the 3 subpopulations. They were smallest in the smallest group, SP2, where the mean carapace length was 9.7 centimeters, medium in SP3, with the intermediate size clutch or carapace length of 10.1, and largest in SP1, where the mean carapace length was 10.9. But even after we accounted for the effect of body size, SP1 still had significantly larger clutch sizes than the other 2 subpopulations. And SP2 showed a slight increasing trend of clutch size over time. We're unsure if this is actually the individuals laying more eggs or just the effect of new individuals who lay larger clutches entering the reproductive population. So we've had no confirmed evidence of more than 1 clutch per year. The average nesting frequency that we got from the data was 80%, and there was no difference among the subpopulations. However, this is a minimum value. It's very complicated to determine as the turtles kind of move around. They can be missed easily. This is a volunteer-driven program after all, and we can't expect too much of these people when they're out till 2 a.m. So we did do a smaller group of turtles that were radio tracked, pooled them together, and it's believed that they nested in 56 of the 59 years. Turtle tracking years, so that would be 95%. Although a lot of us, through anecdotal observations, do believe it's closer to 100%. On to the pretty graphs. So nesting trends are decreasing. They're getting earlier every year. So the earliest nest start date, so the first nesting turtle, the earliest was May 24th. You can see in the blue graph there in SP2 in 2010. The latest start date was June 27th. You can see in the third box on the right. The latest that nesting ended, the earliest that nesting ended was June 7th, also in 2010 in SP2. And the latest it ended was actually July 14th in SP1, as you can see by that pink arrow. So all of them have been trending earlier over time, and it equates to about 6 to 10 days earlier over the 20-year period we looked at. And the nesting time varied quite dramatically between the subpopulations. You can see quite a lot of variance there over time, but still that decreasing trend. And SP2 nested earlier than SP1 or SP3, and SP3 nested earlier than SP1. Now that we look at emergence trends, it is very similar, but somehow slightly different. The earliest nesting date was somewhere before August 20th. We had little escape holes dug so that the hatchlings could escape, and it was checked on August 20th, and those hatchlings had already left. So we know that they emerged before that date, but we're not sure when before that date. And the latest was after November 4th, both in SP2. So there was, again, a significant trend towards early emergence in all three subpopulations, about 10 or 12 to 16 days earlier. And emergence was significantly earlier in SP1 and SP2 compared to SP3. And we've had some successful overwintering in the nest for hatchlings in 2 nests now. And finally, incubation trends. So the shortest incubation period has been 64 days. Again, it's SP2 setting all the records. You can see there by the orange arrow, and the longest was over 140 days, which actually isn't even pictured on this graph. The first nests laid are not always the first to hatch or emerge, and mean incubation time didn't differ among the subpopulations, although it was a little longer in SP3. And there's been a significant reduction in the incubation period time, so almost 2 weeks earlier over the 20-year data set. We also looked at hatching success. So the lowest hatching success was 6%, and the highest was 92.5%, which actually occurred last year. So we're trending towards higher success, which is actually very exciting for an endangered species. SP1 had higher success than SP2 and SP3, whereas SP2 and SP3 were not significantly different. And for all three subpopulations, success is increasing by 1.7% per year. And in 2 of the subpopulations where there are lakes and lakeshore nests, we looked at that as well, and there was a 10% greater hatching success on the lake. We also looked at recruitment in these 3 subpopulations. So females recruited to the reproductive population. In SP1, there's been about 18 new nesters. 11 of those were marked as juveniles. 1 was confirmed from a protected nest. This is really exciting and sort of a great feat because you have to monitor them throughout their life pretty well to keep track of them. We give hatchlings small marks, but as some of you may know, those grow out and they become faded and difficult to discern. So you have to catch that turtle every so often to refresh those notches and keep an eye on them. In SP2, 11 new young nesters, 10 marked as juveniles, another confirmed from a protected nest. And then SP3, there's been 23 new young nesters, 16 marked as juveniles, but none from confirmed protected nests at this point. And interestingly enough, there's been 17 new recruits between 2007 and 2017, which predates any nest protection program in that population, which means something's going on there and they're producing lots of turtles in that area. So age of maturation, there's no evidence of females under the age of 18 nesting. Most seem to begin nesting somewhere around 18 to their early 20s. And the upper age is relatively unknown. It's, again, you have to keep a pretty close eye on these turtles and know where they're going. The higher age categories here are likely turtles that we discovered in our known nesting sites that have gone through a stage of wandering around, trying different nesting sites before settling on the known area. So discussion here, quick summary. There are significant differences between the subpopulations, which is rather interesting when we look at the spatial scale of this. The subpopulations are within 40 kilometers of each other. So it's not a large spatial scale we're looking at, and yet we're seeing differences in the rates and trends that we're seeing. All are trending towards earlier nesting, earlier emergence, shorter incubation, and higher success, which is all very positive for an endangered species. And we're starting to see some increased recruitment. Our next steps for this project are to analyze these trends in relationship to climate data to see if this is affecting these trends. Further update population viability analysis and evaluate the role of nest protection in this population, as well as examine the success of moved nests. So any nests that had to be moved because of the risk of flooding or anthropogenic issues we have moved, we would like to know if we are affecting or impacting them negatively. And I want to shout out the co-authors of this paper. So Matthew Smith, Jeffy McNeil, Tom Herman, Norm Green, and Trevor Avery. I played a very small role in this paper, but I was very happy to take part. This project's very near and dear to my heart. And all the students and volunteers that made it happen. This is very much a huge undertaking to get this much data over this much time. And we can't understate the importance of long-term data sets. So thank you, and I'll take any questions.
Awesome. Thank you so much, Carter. So we have about 4 minutes for questions, if anyone has any. Amanda.
Hi, thanks, Carter. That was really interesting. I'm curious about this ghost nest turtle. Like, have you ever, like, is it a female? And have you ever, guys, are x-rayed to see what was going on or palpate? Like, does she have eggs? She's just not laying them every year, or do you have any idea what's happening?
I wish Jeffy was here. That's her turtle. She might just have a reproductive issue. She seems to just want to do the whole thing regardless that she doesn't actually have eggs. But as far as I know, she hasn't been examined to see why she doesn't produce eggs. But otherwise, she acts completely as if she does.
Huh. So weird.
Pamela?
Hi, Carter. Great talk. The shorter incubation time, is that a climate change thing? Have you guys looked at the shifting temperatures?
That's actually going to be the next paper, is looking at these trends compared to the climate data. We're just in early discussion on how we're going to approach that now, actually.
Perfect. I'd be really interested to see that paper. Yeah.
We've got a comment in the questions from Alita. This looks like the hypothesized population explosion in Eastern Ontario when the beavers came by in the 1960s, which resulted in emetoidea only being seen on the roads in the 1990s. Interesting.
Very interesting.
Awesome. Okay, well, if there aren't any more questions, I think you can continue to put them in the chat as well, and Carter can answer them. I guess we can move on to our next lightning chat, which is Shane Gross, how I won the Wildlife Photographer of the Year award. So Shane, if you want to share your screen.
Thank you for having me here. My name is Shane Gross. That's gross as in disgusting. And I'm here to talk mostly about this image. So I'll let you know I'm not a scientist. I'm a conservation photographer. And this image of western toed tadpoles in a lake on Vancouver Island won the grand title of the Wildlife Photographer of the Year competition. And I want to talk more about this image, but first, I thought I'd just give you a little bit about myself since I'm new here. I grew up in Regina, Saskatchewan, and always was attracted to the water, whether it be lakes, oceans, rivers, whatever it was. Many years later, I moved to the Bahamas and became a scuba instructor and got to photograph everything from sharks and all the charismatic megafauna, but I also wanted to shine a light on underrepresented species like this almost blind Bahamas cave fish in a blue hole. I also try to turn my lens on harder to see images, trying to bring conservation stories to a wider audience. I've been published all over the world in various forms. This is just one cover of BBC Wildlife magazine as an example. I came out with a book called Bahamas Underwater, which chronicled my 10 years in the Bahamas, and I'm proud to say that we got the book into every school in the Bahamas, and funds raised from sale of the book go to kids' sea camps, taking kids in the ocean under the supervision of marine biologists. Then COVID hit and blew up that whole thing, so I had to stay within Canada and decided to make the pilgrimage out to Vancouver Island. And now I get to dive among kelp forests and see aliens like the giant Pacific octopus. I do work for newspapers, magazines, whoever will basically get my message out there. This is a story on the front page of the Globe and Mail about the invasive European green crabs. These crabs will uproot entire seagrass meadows while they dig for clams. But it's not just a story about destruction, but also the heroes that are out there working to trap the crabs, and they have seen seagrass regrow where they're trapping. They're also studying the crabs and trying to mitigate their damage as much as they can. Okay, on to the tadpoles. So this is a topside view of the lake. I went out there basically for a day of fun with my partner, and we got in snorkeling. That's me among the floating pond weeds. And at first I didn't see much. You know, there was one tadpole here, one tadpole there, but I'd heard that there were, you know, these huge swarms. So with a bit of patience, I started to see more and more tadpoles migrating from the deep part of the lake to the shallow part to feed on the algae growing in the sunlit shallows. By the late afternoon, the lake bed was absolutely carpeted by tadpoles. There wasn't a single square centimeter of lake bed visible in some areas of the lake. And if I stood still, the tadpoles would carpet my camera and carpet me. And for those of you who haven't been carpeted by tadpoles, I highly recommend it. It's quite hilarious. But as a professional photographer, my goal there was not just to have fun, but also to make as good of images as I possibly can. So I worked really hard, took almost 1,000 shots that day, and spent 6 to 7 hours in the pond, hoping to get a special image. And this is the one that seemed to resonate with the Wildlife Photographer of the Year judges. A bit about the competition. It's the most prestigious wildlife photography competition in the world. It's celebrating its 60th year this year. They presented me with a trophy of tadpoles, which is really cool. It was presented to me by Kathy Moran, who's a legendary photo editor, 30 years at National Geographic. Any natural history story you've seen in that magazine was probably her doing the photo editing and contributing significantly to it. The competition is huge. It received entries from 117 countries, all ages, and received almost 60,000 photo entries. And it attracts the best photographers in the world. So just one example is in the very lower bottom right. That's Thomas Peschak, who recently had the entire October issue of National Geographic for his work in the Amazon. And it also has given me a platform to talk with lots of people around the world, including you all here today. This is just one example from getting to talk to an audience in France. But I've done a ton of media, and it's been wonderful to talk about the tadpoles and the threats facing them and all those kinds of things. I've only spent one day in the pond. Oh, do you have a question?
No, sorry, you're running, your 5 minutes is run up, unfortunately.
Okay. I just have one more little quick thing. The reason that I'm here today is because I want to tell more about these tadpoles. And so I'm asking for people to reach out to me and let me know what your story is. I'd like to elevate your story. Here's all my information. Take a picture of it with your phone. Please don't be shy. I'd love to hear from you. Thank you very much for your time.
Thank you so much. Those are beautiful photos. So if everyone could please save their questions for Shane, you're welcome to put them in the chat or save them for at the end. We're going to bring up Teagan Netten, I hope I'm pronouncing that correctly, for engaging community and conservation practitioners in long-term monitoring of Ontario snakes.
My name is Teagan. I am a third-generation settler. I'm presenting today from Toronto, where I live and work on the traditional territories of the Huron-Wendat and Petun First Nations, the Seneca and the Mississaugas of the Credit. I am the Conservation Science Coordinator at Ontario Nature, where I work on community science projects and research projects, including the recently published Ontario Reptile and Amphibian Atlas. And this project I'm going to speak to briefly today, the long-term monitoring protocol for snakes. So, as many of you likely know, snakes are a highly threatened group in Ontario, and there's a lack of baseline population data on most species. And that's partly because snakes are cryptic and pretty difficult to study. And so in 2018, Ontario Nature developed a protocol, a standardized long-term monitoring protocol for snake surveys in partnership with experts and conservation professionals to try and fill that gap and improve our understanding of snake populations in the province. So we currently have over 50 sites across Ontario that are monitoring using the long-term monitoring protocol, the LTMP. Ontario Nature monitors 6 of those sites, and the rest of them are done, surveyed by our wonderful partners. So we have people from conservation organizations, provincial parks, private landowners, universities, volunteer community scientists, all sorts of different people participating in this project all across Southern Ontario. We are still recruiting more partners, so we have pretty good coverage and we have a lot of sites, but we are hoping to get some more, especially in some of those areas where we still have gaps. And so each of these sites has a artificial cover object transect set up, consisting of 24 plywood boards set out in open habitat that is suitable for snakes, ideally edge habitat. And during surveys, people go out and check each cover board, searching underneath them manually and collecting data on any snakes that are encountered. The protocol requires a minimum of 8 surveys each year, starting in early May. So most of our partners are done surveys by mid-July, but there is an option to continue throughout the summer and into September. And on top of the snake data that we collect, we also collect weather variables and some habitat data. So this project has been, I skipped a slide. This project has been successful at capturing 12 of Ontario's snake species. So that's the majority of them, including several species at risk. So these are all snakes that have been found under cover boards, including eastern fox snakes, hognose snakes, Butler's garter snakes, and even a few gray rat snakes. This graph here shows a summary of the number of snakes that have been found in each year of the project. The N's there represent the number of sites surveying in a given year. So as you can see, we've had a huge increase in number of snakes over time, along with an increase in the number of sites. So the program's really grown in the past 5 years. The species at the top in red colors are the at-risk snakes and skinks are also on there. And then the gray and blue colors are more common species. And in total, so far, we have collected data on over 8,000 snake species. So we have a good data set going, but we are hoping to keep expanding that over the next several years. So as I said, we're still in that expansion phase right now. We are still recruiting partners. We're hoping to continue growing the program over the next couple of years. And then we're going to switch to ensuring our partners can continue to survey year after year so that we get lots of good multi-year data from each site, which will be very helpful when it comes time to analyze the data. I'm quickly going to put up this long list of all the partners who have been involved with developing the protocol and who currently do surveys. And then I want to quickly thank also our funders who have made the project possible. Thanks.
Awesome. Thank you, Teagan. Perfect. So if anyone has any questions for Teagan, please put them in the chat or save them for the end. And I'm going to ask Jessica Harvey to share her screen on non-scientific data is still good data, the value of anecdotal information in science.
So I think this actually is something we don't talk about that much, but I think is kind of an important thing for us to consider in our day-to-day and how we approach our work. And so I just thought that I would give you guys my perspective on things. So we've all been in situations when we're at work or when we're doing our research where we come across really, really cool things. And often they're not really scientifically significant. And so we don't really have a way to tell those stories within the realm of what we do as scientists. Often reviewers and supervisors will actually downplay those non-significant findings. Journals have a bias towards publishing statistically significant results. And in some cases, my case as an example, we find ourselves in jobs and roles that just don't allow for the scientific rigor necessary to obtain powerful data, never mind significant results. And just to be clear, this isn't me advocating for the diminishment of scientifically rigorous work at all. Following a clear and robust scientific method is always the utmost goal. But along the way, we often hit constraints that limit our ability to collect such data. And we also have access to supplemental data that, in my opinion, is also really valuable. And I think there was a really nice contrast in our last 3 talks between Shane's naturalist approach to biology versus some of those really great scientific results. Let me just move forward. So where does my perspective come from? So I was one of those kids that was playing with tadpoles and, out there in the world. I went to school to become a scientist, and then I became a professional as a consultant. And so I had this transition from naturalist and outdoorsy to scientist to consultant. But I don't think that made me give up those other traits. And my camera roll is a perfect example of that. It's equal parts my dog and plants and animals. So what do I mean by non-scientific data? So we can have non-scientific results, naturalist observations, point-in-time observations, incidental observations, traditional ecological knowledge, as well as, and all of this counts as anecdotal data or non-scientific data, for lack of a better term. And it comes from personal accounts and observations rather than data collected through scientific methods or systematic research. So how can we use this data? First thing that this data can really help to inform is what we want to apply those rigorous scientific methods to. So anecdotal data is often a really great information source to give us ideas of what we want to question and how we want to build our hypotheses. And a great example of that is that in BC, if you look at the literature, rattlesnake diets were studied long time ago, 1980s, and amphibians aren't on the list of things that rattlesnakes commonly eat. This is an X-ray from my final year of my master's research, and we suspect that that is actually a western toad inside this rattlesnake. And so there's a great research question for a future master's student. What we can also use anecdotal data for is making it matter. And so Shane is a perfect example of this, where we're bringing the science and the community and everything together. So in herpetology, we don't tend to have the charismatic megafauna. Nothing is furry. A lot of our critters will creep people out. And so this can be really useful for connecting people to what we do, creating a narrative around our science with the anecdotal data. And lastly, sometimes we don't have the opportunities to collect really robust data. And so we have to use this anecdotal data or point-in-time data to inform decisions. And this is where we do have to be really cautious. We can use this to inform our decisions, but as a very prestigious herpetologist has in his email signature, the absence of evidence is not the evidence of absence, particularly when it comes to snakes. And so we must be cautious when using anecdotal data to inform any decision making, because we can use it to say yes, an animal is there, but we can't necessarily use it to say no, it's not. And so we must really approach it with a conservative eye and follow Carl's sage advice. And that's it for me. Thanks for letting me weigh in, and I guess there'll be questions at the end.
Yep, perfect. Thanks. Thanks, Jessica.
No
All
worries.
right. So last but certainly not least, Fred Schueler, with when is a starling a baby raccoon? Mistakes in mimicry and Treefrog calling.
Well, maybe I'll just describe what's going on. Treefrog started out on the Bruce Peninsula when we were doing herpetology of the Bruce Peninsula, and we found that there were no either versicolor or great tree frogs on the Bruce Peninsula, the outer Bruce Peninsula. But there was one old record by Don Sutherland where in 1989 he'd recorded some, but he was very rushed and we weren't sure what it was. And one of the things that we've been sort of concerned about is the fact that the calls of baby raccoons, and you can Google calls of baby raccoons and get recordings on YouTube, which I would show if the thing were working, that the calls of baby raccoons are very similar to those of hyloversicolor. And then we came home and we started to hear starlings, and I've got slides that have quotes from my field notes about torrents of broken glass describing the singing of starlings. And through the years, we've heard various amounts of Treefrog-like calls from starlings. Sometimes just one call in the midst of a bunch of their broken glass. They also mimic peepers. And one of the interesting things is that a lot of these mimicries occur before the tree frogs start to call. And so the starling's up in the tree vocalizing, and there's this one call that you could mistake for a tree frog if you didn't know it was a starling. And so I'll just say that that's sort of the thrust of what I've said, and I'll send around the PDF of my slides so people can see them in the future. So I guess that's less than 5 minutes.
Thank you, Fred. Sorry we couldn't get your presentation up on the screen. Okay, well, as that's the last presentation, I'm gonna pass it back off to Hannah to field the end of the conference. Thank you, everybody.